Interstitial perfusion culture system. The culture system consists of two parts, each housing two independent perfusion circuits. Materials of each circuit. All components must be sterilized using low-temperature method. One sterile screwdriver. One debubbler with cap. Scaffold holder, 2 parts and 1 screw. 2 oxygenator tubes with anti-squeeze sleeve. 2 oxygenator tubes. Culture chamber, 2 parts, 1 internal gasket and 1 pump tubing. Multi-well plate with cell-seeded constructs and complete culture medium. Reservoir, 1 cap with 3 holes, 1 air filter. Sterile forceps, sterile gloves. Petri dish and pipette. Before starting, assembly must be done in a biological safety cabinet by 2 operators. The main operator wears sterile gloves. The support operator wears normal gloves. Take a sterile forceps. Take the 2 scaffold holders and the screw. Take the Petri dish. Procedure: assembly of the culture chamber. Take the multi-well plate with a cell-seeded construct. Insert the cell-seeded scaffold in the first half of the scaffold holders. Assemble the second half of the scaffold holder by fitting the holder peg. Lock the holder with its screw using the screwdriver. Take the sterile culture chamber. Insert the gasket in the culture chamber and firmly insert the scaffold holder in one of the 2 chamber parts. Assemble the culture chamber by rotating one half on the other one until the external gasket is completely covered. Procedure: assembly of the culture circuit. Fill the reservoir with 40 ml of complete culture medium. Lock it. (35 milliliters). Connect a sterile filter to the shorter tube of the reservoir. Connect one oxygenator tube to the reservoir outlet on one side. and to the culture chamber inlet on the other side. Connect one side of the other oxygenator tube to the culture chamber outlet. And the other side to the debubbler. Connect the two oxygenator tubes with anti-squeeze sleeve to the pump tubing. One oxygenator must connect the debubbler to the pump tubing. The other one must connect the pump tubing to the reservoir. At this stage, the first perfusion circuit is ready. If needed, assemble the other 3 circuits using the same procedure. Place the culture chamber vertically on its holder support. To obtain filling of the culture chamber and simultaneous debubbling. Lock the culture chamber on its holder support with the 4 support screws. Insert one pump head on the roller pump for each circuit. Operate the pump to fill the 4 circuits. Use a very low flow rate to fill the circuits to avoid damage and/or washout of the seeded cells. Insert each pump tubing in an individual pump head. And lock the pump head on the pump tubing. Now press Start on the pump and the circuit will start to get filled. When the circuit is completely filled and debubbled, rotate the chamber in horizontal position. Insert the culture system in the incubator. Place the roller pump on the incubator top. Close carefully the incubator door in order not to squeeze the oxygenator tubes.